Have you ever wondered how our bodies fight off infections? It's like a microscopic battle going on inside us all the time. One of the amazing weapons our bodies use are things called antibodies. Antibodies are special proteins that latch onto invaders like viruses and bacteria, marking them for destruction by our immune system. Polyclonal antibodies are like a team of antibodies. Each member of the team recognizes and attaches to a different part of the invader. This makes them very effective at neutralizing threats. They are like a group of defenders, each grabbing a different part of a monster. Now you might be thinking, what's the difference between polyclonal antibodies and monoclonal antibodies? It's all in the name. Monoclonal antibodies are like a single soldier, highly specialized and identical clones of each other. They target one specific spot on the invader. Polyclonal antibodies, on the other hand, are a diverse group, each member with its own unique way of recognizing the target. Why are polyclonal antibodies important? They are incredibly useful tools in medicine and research, from diagnosing diseases to developing new treatments. Polyclonal antibodies play a vital role in understanding and improving human health. So how do we make these amazing polyclonal antibodies? It's a fascinating process that involves harnessing the power of the immune system. Here's a simplified breakdown. Step 1. Immunization. First, we introduce an antigen to a healthy animal, usually a rabbit or a goat. The antigen is like a wanted poster. It shows the animal's immune system what to look for. Step 2. Antibody production. The animal's immune system responds by producing antibodies against the antigen. Remember, these antibodies are diverse, each targeting a different part of the antigen. Step 3. Collection time. After a while, we collect blood from the animal. This blood is now rich in polyclonal antibodies specific to the antigen we introduced. Step 4. Purification. The collected blood is then processed to separate the antibodies from other blood components. This ensures we have a pure and potent batch of polyclonal antibodies. Polyclonal antibodies are like versatile tools in a scientist's toolkit. They have a wide range of applications in both diagnostics and research. In diagnostics, polyclonal antibodies are used to detect the presence of specific substances in biological samples, like blood or tissue. For example, pregnancy tests use polyclonal antibodies to detect a hormone called HCG, which is present in the urine of pregnant women. Polyclonal antibodies are also essential in medical research. They can be used to identify, purify, and study proteins involved in various diseases. By understanding how these proteins function, scientists can develop new drugs and therapies. Furthermore, polyclonal antibodies are valuable tools in studying the immune system itself. They help researchers understand how the immune system responds to infections and develop vaccines for various diseases. Section 4. Advantages of polyclonal antibodies. Polyclonal antibodies offer several advantages that make them highly valuable in various applications. First, their diversity is a major strength. Because they recognize multiple epitopes on an antigen, they provide a robust and reliable detection method. It's like having multiple witnesses to a crime. The more, the better. Second, polyclonal antibodies are relatively inexpensive and quick to produce compared to monoclonal antibodies. This makes them a cost-effective option for many research and diagnostic applications. Third, polyclonal antibodies are highly stable and have a longer shelf life, making them easier to store and transport. This is especially important in resource-limited settings. Section 5. Disadvantages of polyclonal antibodies. While polyclonal antibodies offer numerous advantages, they also have some limitations that scientists need to consider. One limitation is batch-to-batch variability. Because each batch of polyclonal antibodies comes from a different animal or even a different breed from the same animal, there might be slight variations in their specificity and affinity. Another limitation is the potential for cross-reactivity. Sometimes polyclonal antibodies might bind to molecules other than the target antigen, leading to false positive results. Finally, while polyclonal antibodies are excellent for detecting the presence of an antigen, they are not as suitable for applications requiring highly specific binding, such as therapeutic interventions. Section 6. The future of polyclonal antibodies. Polyclonal antibodies have come a long way since their discovery, and their future looks bright. Scientists are constantly developing new techniques to improve their production, purification, and application. One exciting area of research is the development of recombinant polyclonal antibodies. These antibodies are produced in the lab using genetic engineering techniques, offering greater control over their specificity and affinity. Another promising area is the use of polyclonal antibodies in nanotechnology. Scientists are exploring their potential in developing targeted drug delivery systems and biosensors for various applications. As our understanding of the immune system grows, so too will our ability to harness the power of polyclonal antibodies for the benefit of human health.